What we're going to do today is I'm going to give an introduction to the context of not so much gene patenting in general, although I'll touch on that, but on the case that I think most of you probably came to learn about. We're all neck deep in following the case that was brought about genetic testing for inherited susceptibility to breast and ovarian cancer. And we'll be looping back to that, and that will be the centerpiece of what we discuss today. But here we are on Capitol Hill, and this is a judicial matter, so why are we here? And I just wanted to bring to your attention, the reason we thought it would be useful to do a briefing in this venue is because all three judges, for example, in the most recent ruling from the Court of Appeals for the Federal Circuit, all three judges had explicit sections of their discussion devoted to the role of Congress. There have been three bills that are directly pertinent. Lynn Rivers, when she was a member of the House, had a bill in the, I think it was the 110th Congress. And then Representatives Becerra and Weldon had a bill that was considered in the last Congress. Representative Wasserman Schultz had an amendment to the patent reform bill that actually was passed. It became Section 27. I actually don't know. Some of you may well know much more about that, and if you do, please come talk to me because I want to know the backstory of that amendment. I don't actually know the whole story there. One thing just to flag it, I misspelled her name. I forgot the T in Schultz. So it's back in there in the briefing, but I did notice the error, but I didn't notice it in time to fix it for the briefing today. So I'll fix it before we send it out any further. So obviously some of these issues may loop back to Congress in a very tangible sense, in the sense of perhaps statutory change or proposed statutory change. But really, the reason the reason we're doing a briefing is because we thought you'd be interested. This is just a hot case about hot science in a really controversial area. Our role today is mainly descriptive and explanatory. The three of us on this panel don't actually agree on things, and this may actually become apparent, and if so, that's great. But our purpose today was to try to lay out what has happened, why has it happened, and set the context for this debate, because we're not at the end of this story, and what we would like to do is lay out the kind of the groundwork for what's left to debate and where are the remaining uncertainties and controversies. And in light of that, one of the two things to say about the role of the briefing today. One is that we will be available. There will be an email that is sent out to those of you who have come and to many other people who couldn't come. We are full disclosure, we are taping this session, and we aren't showing it live, but the reason we did that is we thought some people who couldn't be here would like to be able to see it, so we are going to post this after we've done all the logistics of making it available. It'll be posted on the web, we also have a briefing page that has a lot of the background documents, including the file wrapper that is the prosecution history of the key patent, patent 5,747,282, which is the composition of matter patent that still is the one that is being discussed at the Court of Appeals level. So the background documents are there in public forum, and a lot of blogs are available. Last thing to say about that is I've done politics. I've actually worked for 2 congressional agencies that have been killed in the legislative process. I had a grade on the Office of Technology Assessment and a little bioethics committee that existed for exactly 1 year, never got any work done, and died in an abortion crossfire. So I've worked in abortion politics, tobacco politics, cancer politics, and mental health politics. All of those are intense political areas. But I have to say, so is patenting. The ideology is really, really strong, and the rhetoric is vitriolic. One thing that the three of us on this panel, I think, share is a commitment to trying to focus, constrain the range of the debate within the zone of places where we can find evidence and strong argument, and conduct the debate in a civil manner. So trust in the kind of democratic politics they write about in the Federalist Papers. That's actually the reason that my colleagues here were invited to be the other panelists. So now I'm going to shift to substance, and I'm going to do two things. I'm going to lay the context for gene patents in general, and then turn a little bit to this particular case, although Kevin and Arti are going to go into much more detail into those issues. And I actually think a lot of the most interesting things are going to come when you guys have a chance to ask us some questions. So the rules are, I'm going to do an introduction. Kevin's then going to speak. We've changed the order because we looked at what we were going to talk about, and it seemed more logical to go through the order that we're now going to do, which is me, and then Kevin, and then Arti. You've got bios for all three of us. I don't want to spend the time to repeat all that stuff, except to say the following. I'm here mainly because this is the sort of stuff we study under this grant. Kevin is here because he writes a blog. This is the main reason he's here. He writes a blog that has done an immense amount of work of getting information out about all the cases that are hot in biotech, but in particular this case. And the Patent Docs website is the place where you can go to get all of the amicus briefs for this case and others. And the rules on the Patent Docs blog are no ad hominem attacks, although it leaks out every once in a while in the feedback mechanisms, the comment section, but even those are moderated, so I don't know what you cut out, but probably a fair amount.
A fair amount.
And then finally, Arti is here because she is one of the most distinguished professors in the U.S. that deals with biotech law. Her case book has just come out, or is coming out. Is it out?
It's a collection of seminal articles.
Seminal articles,
yeah.
Not a case book,
Yeah.
excuse me. See, I'm not a legal scholar.
But one of your articles is considered seminal by me.
So, and Arti has the distinction of also having worked in the U.S. Patent and Trademark Office, and she will be commenting a little bit about the role of an executive branch in the mediation of these controversies. So, what I'm going to do is I'm going to show just 3 slides that show some data. So if you could give me the first of those slides. This is what we're talking about in general, and it's a really messy slide, but the only thing you need to do is grok the blue stuff, the blue parts of the graph. I'll do the counts for you. As of last week, there were just shy of 60,000 patents that have been issued by the U.S. Patent and Trademark Office that mention RNA and DNA in their claims. And we've called those things DNA patents as a shorthand. If you do in your brain, if you think it costs something like $16,000 on average to get a patent, we've got about $1 billion of stuff. People have paid about $1 billion to get the rights that are represented in this slide. 60,000 sounds like a lot, but the number of patents that are actually relevant to what we're talking about today is very small. And the nature of what is claimed in those patents is highly, highly, highly variable. So when people use the term gene patents, it's a very, very fuzzy term. So what we're going to be talking about today is primarily claims on DNA, either DNA molecules or methods for extracting information from DNA molecules in a diagnostic sense or a forensics or whatever. And it's a very small subset of the number of patents that have been granted on DNA or RNA. And what characterizes them is they say something about making, using, selling, or doing something with a human gene. That's mainly what we're going to be talking about today. But to tell you the truth, there's not a soul on the planet who knows how many patents fit under that category. You can get a kind of a feel for it by doing very specific case studies in a particular instance, or if you're looking at a particular domain of research, but it's very hard to actually get a global feel for what's going on. But what we know is it's a very small subset. So that's one. The next slide, if we could do that. This is a collection of all the litigation that Chris Holman at the University of Missouri, Kansas City Law School pulled together for an article he published in 2008 in Science. And this tells you who has sued whom over a gene patent. And it's in 4 categories. And what you will notice is we've broken it down into how many cases have been brought in total and how many have gone to which stages of litigation. And what you'll see is the only ones up until he had done his census, this predated the case we're talking about today. The only cases that had actually gone to a trial and a decision and set any sort of case law precedent were the cases in the therapeutic protein and a couple of the research tool cases. But there were 0 cases of diagnostic testing or forensics, which is kind of a similar, shares some similar features, that had gone before a judge and where somebody had to make a legal decision. So there's been the point here is interesting. Where there's been litigation, it's been about DNA as a way of making something valuable, like a vaccine or a therapeutic protein that's very expensive, but the DNA is a step in a long process where it's fairly obvious the DNA is kind of a piece of engineering that's used for a process that involves a lot of human ingenuity. Diagnostics is a completely different context, and that's the case that we're now going to be turning to, but we have one more slide. If we could show that. This is, again, a slide that it's in your readings. I didn't expect you to be able to do it. You only need to notice one thing about this slide, which is there's a big bar at the left edge of each of these slides, and that's BRCA. And what we're counting here is the number of references either in English language print media all over the world, so US, UK, Australia, New Zealand, etc., newspapers saying something about a gene patent controversy. That's the bottom one in green. And at the top is mentions of cases that were controversial that have made their way into reports. That is, policy reports for the Danish government or the US government or the UK government or for the World Health Organization or whatever. And what you notice is that the case we're talking about today comes out of, all of this predated the case we're talking about today, all of these citations. But the breast cancer testing controversy has been the hottest one in gene patenting. So just to note the anomaly, litigation's been mainly about therapeutic proteins, didn't elicit a whole lot of public controversy or policy response, but diagnostic testing has elicited a lot of public comment and controversy, but very little litigation. And this is the first really big case that's been really focused on that question. So we're now done with the I'm a professor, you have to look at my slides part. And let me lay some other features that are distinctive about this, and then I'll turn it over to Kevin to take it to the next level. Just to finish up the thing about what's distinctive, one is that this is the case. This is the first case that's come before a judge, as I said, but many other gene patent cases have actually already gone up to the level of the Court of Appeals for the Federal Circuit. By my count, there are 11 that have been decided that are about gene patents, and in every case, it was just kind of assumed that, of course, you can patent a gene. Of course, you can patent a DNA molecule. That's been true not only in the United States, but in all the other jurisdictions where it's been tested in Europe, European countries, and in Australia, New Zealand, and Japan, etc. So yet we have this case. One of the other things that's interesting about this case is the nature of the litigants. This is not your usual patent case of Company A suing Company B over who's going to make money off of this technology. This was a case that was sponsored, organized, and coordinated by the American Civil Liberties Union and the Public Patent Foundation, who are focused on policy concerning gene patents. That's why this case was raised, and it's distinctive. The nature of the litigants is also interesting. The plaintiffs in this case were in 3 general groups. One was women who wanted to get tested, and the policies of Myriad Genetics, the particular owner of the exclusive rights in this case, they were alleging that the policies of that company were getting in the way. And the second class was people who run laboratories or run services that do clinical diagnostics. And third was organizations that represent those health professionals. So those are the kind of the 3 classes of plaintiffs. Only one subclass of a subclass was left standing at the end of the Court of Appeals for the Federal Circuit. So I'm sure we'll get into that inside baseball as we discuss the case. But it's a different kind of plaintiffs. And this is a case that is basically, we would like to infringe, but we don't want to incur infringement liability if we do that. So it's kind of funny. Usually you have somebody who's already got a product in the market, you sue them afterwards, and you fight over the damages. Here the damages haven't happened yet, and it's a matter of policy for the most part. It's a very unusual case in that respect. It's also unusual in the level of attention. There were about a dozen amicus briefs at the district court level, in the Manhattan District Court, where this was decided in March of last year. And then there were 29 amicus briefs at the Court of Appeals for the Federal Circuit level, which is a lot. One of those was particularly significant, and that was the one that was filed by the U.S. government and was defended by the Solicitor General himself at the oral arguments on April 4th. So here's where the U.S. government has staked out a position. It's interesting in two respects. It's the first time Solicitor General's ever been before this court, and it's interesting because there are actually two positions within the federal government that are not completely in alignment. One is the position of the U.S. Patent and Trademark Office, which is part of the executive branch, and then the Department of Justice and the brief that was filed by the U.S. government for the government as a whole. So there's some dissonance within the federal government still going on, but there is an official position of the U.S. government that was basically, you can't patent DNA across the board. You should not be able to patent DNA that corresponds to a sequence that would be found in nature, but you can patent DNA if it has been engineered or changed or would not be found in nature. So it was pretty close to the decision that was actually defended by Judge Bryson in his arguments. So that's kind of sets the context for what we're going to be discussing, and I have lots of opinions about what should and can and should happen, but actually I don't want to spend the time going into that. We can handle that offline, and I would like now to turn it over to Kevin.
Thanks, Bob. Thanks for coming, everybody. Can I have the first slide of mine? I apologize to anybody who understands patenting, but what I found, what you have to realize is this case came upon the patent bar kind of like a hurricane. We're not used to the kind of attention that the ACLU brings to any topic, and even some of the, it's a very arcane part of the law. Bob talks about the blog that I write, but that's recreational as a day job. I'm a patent lawyer. So, you know, we do it, my partners and I do this in our free time if we have any. So I just want to go through a little bit of what patenting is, because a lot of people have misunderstandings that have come out in this case about what a patent does. First of all, it's a property right. It's in the Constitution, but it's very limited, and it's limited, I think, in three critical ways. One is it's limited in time. The right only extends for 20 years from your first filing date, which means it expires. It's not perpetual. It's not even like copyright that goes on for 75 to 100 years. It expires. Secondly, it's not a right to do anything. It's a right to stop other people from doing things. And that sounds like it's a quibble, a lawyerly quibble, but it really has a great deal of significance, because it means that all you can do is be in a position to negotiate licenses, et cetera, with people who want to practice your invention. And the other thing is it only gives you the right to sue someone, and occasionally, it used to be much more than occasionally, get an injunction, and that's become much, much harder. It's really about money damages, and Bob pointed out that a lot of the cases have been company to company, because that's usually the context in which these disputes arise. The claims in a patent, again, getting even more esoteric, the claims are what define the property right. If I can have the next slide. It's kind of like the boundary to your house. You know, the idea is that just like you have a boundary line on your property, the claim defines what the metes and bounds of your right to exclude would be. And the patent not only has claims, it also has disclosure. And there's what we call the quid pro quo, which is you have to disclose adequately so that someone with skill in the art can actually practice the invention once it expires. Because once it expires, the public has to have the right to practice that invention fully without any recompense to you in perpetuity. So that is the bargain, I think, that between the government and individuals when they give patents. Can I have the next slide? Okay, did we miss a slide? I think there's one more thing I wanted to say, but I may not be right. No, that's good, okay. So before we get into the myths, let's talk about the case just briefly. There are two types of claims that are kind of at issue. Keep in mind that there are, I think, either 5 or 7, I get the number mixed up, patents that Myriad has licensed from Utah or has on its own. and only a certain number of the claims of the patents were actually at issue. And the two types of claims are what we'll call the composition of matter claims, the DNA itself, and the method claims, the diagnostic method claims, and we'll talk about that at the end. It's interesting to a patent lawyer because I think that it's unlikely that the composition of matter claims, at least most of them, would be infringed by anybody who actually practiced the diagnostic method claims that are kind of at the heart of why people are upset about this case and maybe at the heart of what the plaintiffs say they want to do. That's because historically these claims are written sort of like therapeutic claims in the sense that they claim not only an isolated DNA, but an isolated DNA that encompasses the entire BRCA1 or BRCA2 gene. Very important if you want to make that as a protein, because you have to have the whole thing in order for it to work. The way that diagnostic methods are practiced, especially these days, is I think it would be very unlikely for anybody to isolate the entire gene to do the sorts of tests that want to be done. So again, one of those disconnects for a patent lawyer is why did they pick these claims and leave out several claims that arguably, even if they won, totally would still be infringed by anybody who is practicing the claims and still give rise to a lawsuit. But as Bob said, this is a very political case, and there's a bunch of allegations and statements that have been made that, in my view, aren't true. And one of them, the one that's the most visceral, the one that gets people really upset, is this ownership myth. Michael Crichton kind of famously wrote an op-ed piece in the New York Times a few years ago where he posited a patentee ringing your doorbell and handing you a bill because your liver was using the patented gene. The ACLU, of course, has their little logo here that they've put in most of their mailings and have little buttons. I have one. And then there was this idea of who owns you. Well, anybody who lives in America should realize the 13th Amendment means nobody owns you. And all around the world, there are also prohibitions to anybody actually owning a human being. So what is patented, what is claimed, is very different from owning a piece of any of us for a lot of reasons. The information myth actually was very, very powerful in the district court because one of the things that DNA is information. It's an array of molecules, of nucleotide bases that human beings interpret as having a sequence that then encodes a protein. That exists as a chemical whether human beings understand it or not or appreciate the information or not. But the judge was convinced that DNA being the physical embodiment of genetic information made it different from other biological molecules, and he's right as far as that goes. The problem is that the information isn't patented. You can take any patented gene and take just the sequence and interrogate a database or design another gene that would be related to it, and none of that's infringing. You only infringe when you actually have the molecule itself isolated in a test tube. Can I have the next slide? Then there's the inhibits research myth, and this was one that has started probably more than a dozen years ago by an article in Science that talked about the tragedy of the anti-commons, which you prevent basic research by having patent rights to genes. Well, it hasn't turned out that way, and one evidence of that is if you look at PubMed or one of those databases, there are more than 9,000 scientific journal articles that have been written about the BRCA genes since the patents were granted, and that's because the myriad and no gene patent holder cares about basic research. In fact, it will probably help them. Commercial research, where somebody's charging patients to give them their genotype or their phenotype, that's something that is commercial activity that the people who are doing it are charging for, which is very different from basic research. And that, obviously, you might imagine, raises hackles. I would also say that, conversely, the fact that a patent requires that there be this disclosure of full disclosure of the invention is something that isn't always required in scientific journal articles. In fact, when the Human Genome Project results came out, the Patent Office made a decision that they were not going to allow some land grab of genes just because you happened to notice that there was a gene sequence that you could identify. You actually had to provide the public with some use for that gene. You had to know what it did. And there were many people who filed patent applications before this rule came down who found, to their chagrin, that they didn't have the requisite knowledge of what the gene did to satisfy the patent statute. So I think that there's a disclosure issue element here as well. And finally, there's the natural product myth. You can read case books that say natural products can't be patented, which really isn't true because I could give you a list of natural products that have been. So I don't think there's any legal precedent that says per se a natural product can't be patented. But even if there were something like that, isolated DNA, not only in my opinion, but in the opinion of at least 2 of the 3 judges on the Court of Appeals, is not a product of nature, and cDNA, which I'll talk about in a minute, a little piece of scientific jargon, certainly isn't. Can I have the next slide? So genomic DNA is essentially this sort of piece here that I have just snipped out of the chromosome. When I say just snipped out, it takes a lot more effort than that. But the difference is really that you've taken the DNA as it exists in nature and you've extracted it from not only all of the other proteins and cellular components, but from all the other DNA that exists in the cell. And given the fact that there are, millions of billions of base pairs of genes in a cell, this at one time before the Human Genome Project was almost a Herculean feat. There were graduate students who spent their whole careers doing this. cDNA, on the other hand, is a product of human invention in the sense that it can only be prepared by a scientist who has isolated a molecule, an RNA molecule, from a test tube and enzymatically converted it. And if I can have the next slide, it's just a graphic of that. The top line is the way the gene looks in a chromosome. Human genes and most eukaryotic genes are different than bacterial genes in that the important parts, the parts that encode the protein, are separated by varying amounts of things that are nonsense, that don't encode anything. And the cell snips those out when they're actually processing the gene and transcribing it and translating it into protein. That third line is what's left when you do all of that. And that's a different molecule, as Bob said, RNA, that then has to be enzymatically converted into DNA that's what is claimed. And so and this was a decision that formed the basis for two of the judges, actually all three of the judges, deciding that cDNA had enough human intervention, had enough hand of man, if you will, that would be patentable because it was sufficiently different. Just to give you the scorecard of what happened at the Federal Circuit, as to the claims to cDNA, as to these claims that things that were invented or produced or processed by man, all the judges believed that that would be patent eligible, as did the Department of Justice, because that was something that showed enough. Where two of the judges differed from the third, from Judge Bryson, is that the top line is it enough to actually extract the DNA and change it? And the basis, can I have the next slide? The basis for that decision was that the court found that isolation physically changed, and they were right, physically changed the way the gene was in the chromosome as opposed to the way the gene is in nature, and that physical transformation was enough. Judge Bryson didn't believe it was, in part because I think he was concerned, and he said in his opinion, that this would inhibit whole genome sequencing, the fact that you can take DNA, the entire DNA, not isolate anything, and yet today we can target specific genes and sequence them. I know that Bob and I disagree with this, but I think that I don't think that's a problem, because I think that whole genome sequencing, which involves just taking the entirety of the DNA, wouldn't infringe any of these claims because there's no isolation. The thing I want to leave you, the policy thing I want you to think about, is that the court, because it was the question before them, made this decision that isolation, this chemical changing, whether it was by snipping out the DNA or by chemically converting the RNA, made the claims in front of them patent eligible. There was an implication that mere purification would not. And the thing that I'd like you to think about a little bit is that may not be the best decision. They didn't say that, it's not a holding, but it's the implication of what they said. And human beings aren't as smart as we like to think we are. Most of the best drugs in the world are produced by plants to protect them from insect predation. And a lot of those drugs are only purified, if you want to say merely purified, from nature, because the amount of change you have to make to them is to take them from their natural environment where they're either useless or we would require every tree in the Amazon to be processed into leaves for us to chew to get the drug out, or some drug company goes down and actually figures out what the molecule is and purifies it. So I think we have to be careful in the traditional drug context, and even especially in the biologic drug context, because that does implicate a molecule that would be identical to a molecule that exists in your body. If you could in fact make an exact replica of that, why would that not be eligible if it could be a pharmaceutical? Not sure. But the next slide talks about the other claims, and that is these diagnostic method claims. And I'm going to end with two slides on that. And one of them is that they have a structure, and I'll credit Kevin Collins, who's now at Wash U, with this sort of intellectual framework. All the claims are in some sort of a detection or determination step. You do something to get to a biological sample or something, and you get, really, unlike most other method claims, you don't get a thing, you get information. And in this context, you sequence a gene and you determine whether at one area or the other you have a mutation that people have found correlated with disease, and then that inference or conclusion is used to do what the claim is intended to do: diagnose a disease, diagnose a risk for a disease. And so this is really, in many ways, one step removed from the very practical methods of making and using tangible inventions that were the basis for the patent system. Now, we could, if we had more time, we can talk about lots of other examples outside the medical context where these sorts of claims exist and nobody cares, but obviously the implication of human beings makes it important. So I can have the last slide. I think that the importance here of these is that they protect commercial development of the invention. And I know that a lot of times we get to what I think is the beginning of the road. We find the correlation. We find that this mutation in the BRCA gene can predict with 95% certainty that someone will get breast cancer in their lifetime, way higher than the frequency is if they were just part of the general population. But that's really the beginning, in the sense that to take that laboratory evidence, take that correlation, and actually do all the work of making sure that you're always right, because you don't want to give somebody a false negative or false positive in this. So there's lots of things that are very tedious that have to be done, that university scientists don't have the time, in fact shouldn't be doing, that have to be done. And then there's all the practical things, educating doctors and getting genetic counselors that are going to be able to explain this to people, because you don't want people to hear, I have a gene mutation, and decide that they're going to kill themselves. Getting Medicaid and other providers to pay for it. This is all work that we don't really see, but in the absence of that work, you're really not going to get translation of the basic scientific correlate into something useful for people. And the other thing is that there are alternatives to patenting, which requires disclosure, that don't. And I'll posit to you that most of the diseases that people have are not as clear-cut as BRCA. It's not one mutation that gives you a disease with 95% certainty. There are going to be a lot of different genes involved. They're going to be changing either mutations or changing expression levels and such. And the part that we've done that sounded so difficult, the Human Genome Project, is really somebody takes your car and puts all the parts out on your garage floor and then says, okay, now build the car. We don't really know how all those parts go together and how they go together in disease. And it seems to me that what's going to happen over the next 50 or 100 years is figuring that out. And because it's complicated, one of the rules of patenting is that you need a patent if something is easy. You really need a patent because somebody can just copy it. But for something that's complicated, these alternatives, trade secret, exist where you can just not tell people how you get the answer. And it's not necessarily required you tell people how you get the answer. You just provide a diagnostic that gives them the answer with sufficient confidence that it's valuable. And I think that is contrary to the last 30 years of very open disclosure, the academic, industrial sort of marriage of doing basic research in the university, which is what they do, and translating into useful products for the rest of us, which is what industry does. And I think it would be a mistake to do that. That's my policy answer on this. But I think we all have different views on that, and I'm sure we'll talk about them. Thank you.
So thanks first to Bob for organizing this event, and thanks to both Bob and Kevin for doing a great job of laying out the basics of the case and why it's important. I'm going to be talking about institutional questions, in particular the role of the executive branch of the government. Before I begin that, though, I do want to note a caveat. Bob has mentioned that I was a political appointee in the Obama administration. What I'm saying here is based entirely on publicly available information. So there's nothing here that cannot be traced back to a public document. In fact, I'm writing an article on some of these issues, and one of the axioms of writing articles is that you have to cite to publicly available information. The article, by the way, will be coming out in the Duke Law Journal in a few months. I didn't provide it here because it's 20,000 words, and it's probably much longer than anyone would want to read for a briefing, but it will be available in a few months. So, as I said, I'm going to be talking about the institutional roles here, focusing on the executive branch, but not exclusively. Let me start by asking what the executive branch means precisely, because it's not entirely clear in this case, as Bob has already suggested. At the district court level, the executive branch was mostly represented by the PTO. The Justice Department technically represented the government, but the PTO was the main actor. It was a defendant, actually, at the district court level on some constitutional claims, and successfully got those claims dropped. But in so doing, made it clear, the PTO that is, via the Justice Department, that its position was that claims to isolated DNA, which encompass also cDNA, but also the larger category of isolated DNA, represent patent-eligible subject matter. So the PTO pointed out that not only had it issued lots of patents in this regard, but its so-called utility guidelines, to which Kevin briefly referred, which issued in 2001, had specifically stated that isolated DNA, and they didn't even think about the distinction between gDNA, genomic DNA, and cDNA, because it was just all patentable eligible subject matter. So with the PTO dismissed from the case at the district court level, the executive branch actually didn't even have to participate at the federal circuit level. There was no need. It did participate, however, as an amicus, as we all know, and it did file the most important brief in the case of the 29 amicus briefs. Just a side note, the Justice Department has a terrific record before the Supreme Court. Whenever it's participated in a patent case before the Supreme Court, at least since 1996, it's won with one exception. That was the Bilski case, which we can discuss.
We don't know who won that one.
We don't know who won that one, exactly.
No, we don't.
So this
don't.
is,
Offer them the wine.
for purposes of thinking about, if this case does go to the Supreme Court, for purposes of thinking about who's going to be really influential, you should think Justice Department. So at the federal circuit level, the Justice Department didn't have to file a brief, but they did, and they filed it also on behalf of the executive branch. But the executive branch here seemed to be a little bit different. The PTO is part of the executive branch, was listed as one of the agencies represented, but the brief said that the PTO's decades-old policy of granting patents on isolated DNA was incorrect. So let me just quote very briefly from the first page of the amicus brief. It contains a key paragraph about exactly who the appellate staff at the Civil Division of the Department of Justice was representing. The SG did appear at the federal circuit level, but this is a brief written by the appellate staff of the Civil Division. So, quote, "The extent to which basic discoveries in genetics may be patented is a question of great importance to the national economy, to medical science, and to the public health. This appeal consequently implicates the expertise and responsibilities of a wide array of federal agencies and components, including the Patent and Trademark Office, the National Institutes of Health, the Antitrust Division of the Department of Justice, the Centers for Disease Control and Prevention, the Office of Science and Technology Policy, and the National Economic Council, among others." So we're talking about a huge number of different components of the government thinking that they have a stake in this. What's particularly interesting is this is not the first time that DNA patenting has elicited great interest from across the executive branch. In fact, I think it's fair to say that there's been a 20-year history of very strong NIH interest in this question. In the early 1990s, not too long after the PTO began issuing patents on isolated DNA, including cDNA, but also genomic DNA, the National Institutes of Health itself began thinking that maybe it should file some patents on DNA. So it specifically thought, under Bernadine Healy, the then director in the George H.W. Bush administration, that it might want to file some patents on short pieces of expressed DNA known as express sequence tags. Even though it didn't know the particular biological function at the time, it thought that maybe it should file some patents. Ultimately, in the mid-90s, when the Clinton administration came in, NIH decided to withdraw those patents. And then by the late 1990s, the Clinton administration was strongly objecting to the PTO's proposal to grant these patents. So I would differ a little bit with Kevin in terms of the history of how the PTO decided that it wouldn't grant patents to genes of unknown biological function. It had said, and John Dahl in an article which came out at the same time as Becky Eisenberg's article about the tragedy of anti-common, specifically said that, yeah, we're going to grant these patents and there'll be cross-licensing and life will be good. Well, NIH wasn't going to take their answer, PTO's answer. They kept on pushing, and Bob probably knows a lot of inside baseball about this. I can tell just from the public comments, which I've now read all of, that NIH was the main actor pushing and pushing and pushing. Ultimately, it carried the day. In 2001, the PTO issued utility guidelines effectively saying that gene fragment patents to fragments of unknown biological function wouldn't issue, and they didn't for the most part. And in fact, this was one of the key factors. I think this is a little emphasized piece of the debate. So Becky Eisenberg writes her article, Tragedy of the Anti-Commons. Everyone thinks EST patents are going to issue. They don't issue. Turns out, as Wes Cohen, our colleague at Duke, has found, one of the reasons that industry actors say they were much less worried about an anti-commons was because of the utility guidelines. Well, who was responsible for that? It was NIH, Becky Eisenberg, others who really pushed on that. So I think it's a little misleading to say that her prediction didn't turn out. It didn't turn out in part because she changed the course of history through her article. So I include that article, by the way, in my Seminal Articles book. Now, interestingly, NIH didn't talk about patentable subject matter when it filed its comments. Some other folks raised the issue of patentable subject matter, but quite frankly, I do administrative law in addition to other areas of law, and comments that really matter are from major interest groups and from other parts of government. The people who raised these didn't have the clout, really, to get the PTO to pay any attention, and so the PTO sort of, as Judge Bryson put it in his concurring and dissenting in part opinion, their discussion of the whole patentable subject matter was, quote, perfunctory, unquote, which I think is accurate. Nobody really thought, patentable subject matter, why aren't these, of course they're patentable subject matter. So that's back in 2001. Fast forward to 10 years later. Well, in some ways this is deja vu all over again. NIH, among others, is concerned now that patenting mere isolated DNA could create a tragedy of the anti-commons problem for whole genome sequencing, which is the future of how diagnosis and research is going to be done. Now, we can get into questions about whether their perception is accurate. As a technical matter, I think it's fair to say that the problem of potential infringement is clearest with respect to some claims, including some Myriad claims that are on very short sequences. So Myriad has claims to 15 MERS, 15 nucleotide sequences of both breast cancer genomic DNA and cDNA. Chris Holman has gone through this exercise with respect to human gene patent claims in general, and there are not that many, at least in the subset of human gene patents he looked at, that many claims to these short sequences, but there are some. And I think those are the claims that are worrisome for whole genome sequencing. It's the 15 nucleotide sequences, any gene sequence encompassing this 15 nucleotide sequence. I think for whole genome sequencing, that could be a problem. And Bryson's dissent in the Myriad opinion specifically focuses on these 15 MER claims in the Myriad case. Now, as he points out, they're probably invalid on other grounds. In fact, Bob has written an article, a nice article, indicating that they're probably not novel if they really purport to cover 15 MERS and any gene sequence that could include that 15 MER. But as Bryson also points out, that's a very time-intensive exercise. I'm sure Bob can tell us how time-intensive it was to figure out how many different places in the public genome database those 15 MERS were found. So it's much quicker if one just says gDNA is not patentable, and those 15 MER claims would encompass gDNA, so those 15 MER claims are out on patentable subject matter grounds. And the executive branch's position, by the way, would take out those 15 MER claims as well for that reason. And it's a very simple, clean, you don't have to look at the details of the claims say. By contrast, the executive branch position would preserve the what many people consider the core cDNA claims that are most important for therapeutics. I think the reason that we see all this litigation about therapeutics is because they're really valuable, and that's where patents really count. The problem with all of this, this interest by agencies like NIH in patentable subject matter is they've arrived quite late in the game. They could have raised these issues way back when, more than 10 years ago. They didn't. And Judge Moore appropriately points out, by the way, she would have decided the case differently if she didn't have to worry about settled expectations. She makes that clear. She appropriately points out, boy, it's awfully late in the game to be raising these issues. And I, frankly, tend to agree with her. It's awfully late in the game. This goes back to the institutional questions. There's a very interesting back and forth between Judge Moore and Judge Bryson, who write really excellent, both of them write really excellent opinions. What weight should we give to these settled expectations? Well, Bryson says we can't really give that much weight because, look, the courts have the right to construe 101. courts have never addressed this issue. All of these gene patent cases that we've dealt with never raised this issue, so we couldn't get to it. And if we don't have the right to address this issue, what we're doing is effectively giving the PTO rulemaking authority over questions of patentable subject matter, which it does not have. Congress has not given the PTO rulemaking authority over substantive patent law, which is a core principle right now of the way that administrative law operates in the PTO context. Moore says, well, Congress has considered this issue. It decided not to do anything about it, which indicates Congress has sort of blessed what the PTO said and settled expectations on top of that counsel in favor of allowing gDNA claims to go forward. But note that that's what tipped her decision. Otherwise, she wasn't so sure. So Congress hasn't given the PTO rulemaking authority is essentially allowing the fact that PTO has been giving these patents out for 30 years, giving the PTO effectively rulemaking authority. And I think that's something interesting for Congress to consider. Two other things, and I'll end on those notes. What role should Congress's failure to pass legislation play in courts thinking about these issues? That's a very controversial jurisprudential question that we can talk about because people think very differently on that question.
But just to double click on that, both the Rivers bill and the Becerra-Weldon bill were cited by the judges, in particular in Judge Lurie's majority opinion. So
Right,
it
and
played
so
it played a role in the
So
thinking.
Congress considered the issue and it decided not to go forward. What
Right.
role should that play in judges' thinking? That's a very interesting jurisprudential question. And then finally, let me also double click on the SG's track record, or the Department of Justice's track record in as an amicus or as a party at the Supreme Court. If this goes to the Supreme Court is going to be watching very closely what the Department of Justice and the executive branch more generally have to say. So that's it.
So let me, I'm going to open it up for questions, but there's one thing that I forgot to do, which is to acknowledge that Jim Evans was going to join us here. Jim was the chair of the task force for the Secretary's Advisory Committee on Genetics, Health, and Society. This is a group that existed that was advisory to the Department of Health and Human Services that actually did a study on this, made recommendations in a report that became public in February and then was published in April of last year. The committee no longer exists. Jim couldn't be with us, but in his honor, I wore my chromosome tie, various DNA ties. But he sends his regrets. I know he's hosting a meeting down in North Carolina as we're holding a meeting up here, and that's why he couldn't be here. The other point that I wanted to have us start with, and then we'll open it up to questions for you. One thing I didn't quite cash out in my earlier comments is, why is it that diagnostics is a different context from therapeutics? And I think the reason that Judge Sweet and Judge Bryson have gone their path, so far 4 federal judges have heard this case, and let's just review the bidding. All three judges at the Court of Appeals for the Federal Circuit level, the level that matters right now for setting precedent for U.S. patent law, have all agreed that cDNA patenting is fine, and that the broad method claims are not fine. They're invalid. And they also completely agreed on one method claim that is a different type. It's kind of basically for an assay. I'm not sure why it was in this case at all. But they've said it's okay. It passes muster. And I don't think we'll hear too much about that particular claim again. So that's actually a fair amount of consensus, even if we stop right here. The zone of uncertainty has actually been narrowed quite a bit to this question about whether you can patent a molecule, the composition of matter claims, on a molecule that corresponds to a sequence that you would be able to find in somebody's body. So why is that different in diagnostics from therapeutics? You can imagine doing that for a lot of things like vaccines. You're taking bugs and you're more or less taking a sequence that was out there and you're turning it into something that you're going to inject in order to elicit an immune response, for example. That might well correspond to something you would actually find in nature, but you're doing a lot of stuff to make it useful, and it's fairly obvious that you're doing something. Diagnostics, what you're trying to do is capture the information that is coming from a sample in a person's body. And I think that's why Judge Sweet went where he did in his intuition. He called it the embodiment of genetic information. And he's saying, yes, there's a difference in the molecules, but it's not a materially relevant difference. And that's where the judges are differing. You've got two federal judges who have said, yeah, I buy that argument, and two federal judges who have said no. The two that matter right now are the ones who voted with the majority on the three-judge panel. And just to finish where we are procedurally, the decision came down in July, last few days of July. Both sides have now appealed not to the whole Court of Appeals for the Federal Circuit, which they could have done, and not to the Supreme Court, which they also could have done, but they've appealed to the same three-judge panel that made the original decision. And Myriad's arguments are very focused on this question of standing that we only talked about tangentially. Turns out that the guy that was the only plaintiff left standing is changing jobs. And Myriad is arguing that we had no beef with him specifically. We'd sent our letters to the place that he worked, and he no longer works there, so there's no evidence that there's an active controversy that will be resolved. So that's what Myriad's arguing. ACLU is arguing, oh, you missed some plaintiffs here that also should have standing, and the associations that represent them that should have standing. And they want to go back and touch on some of the arguments that were made, particularly in Judge Lori's majority opinion, about, and this is where we can turn right now to where Kevin and I almost certainly disagree, which is what this word isolated is doing, because isolated is doing all the work in these claims. And Judge Bryson's first question to the attorney representing Myriad at the oral hearings was, if I do whole genome sequence analysis, am I infringing these claims? And the lawyer, Castañas, said, no, you would not be, and that may or may not be the case. Kevin, you can represent. Kevin actually makes a very coherent argument. This is the way to read those claims, and I think that may actually be the way they would be read.
Yeah, I think that the problem is that if you don't read them that way, and the way that I think that the ACLU reads them is that, and there's, if you squint your eyes really tightly and ignore some of the words in the specification of the claim, you can read them this way. It is that you just isolate the entire chromosomal DNA from all the other things in the cell, and that would be isolated DNA. And the problem, actually, I disagree a little bit, because the word that's doing all the work in the claim is comprising. Because there's this word in claims, comprising, and it means what I say and anything else. And that, how broadly, if you read that word comprising very encompassingly, that would, if that was read that way, that would be the way the claim would read. The problem is that Frederick Nischner in 1880 did that already. So that's not new. Okay, just isolating DNA from cells has been done for 120 years or so, 130 years. And so if you read the claim that way, it's invalid for all sorts of other reasons. So I think that the way I read the claim is that the isolated DNA has enough of the sequence so that you could make the protein. Now, it's broader than the word consisting of, which is another bit of arcana in the patents that would mean just this, because you wouldn't want somebody adding a few trivial nucleotides on the end that don't do anything and say, well, I don't infringe. But if once you get outside the scope of the DNA that encodes the actual gene, I think that you're treading on, if not novelty grounds, you're over expanding the claim, and it would be invalid also for overbreadth, because you don't tell me what else you can put in if you're going to really expand it to that way. Let me do, before I stop, let me do one thing. Yesterday, I think it was, the Federal Circuit denied Appellee's appeal for rehearing en banc. Appellee's rehearing en banc. So the ACLU's rehearing that the majority made a mistake on the substance, and that these other two groups of plaintiffs, or two plaintiffs, should also have standing. That appeal, that rehearing appeal, has been denied. They have not spoken to the standing one yet. So it suggests to me, reading tea leaves, that either the court is going to grant that or is at least thinking about it, because it's a different argument than as legalese as it may be. It really is the critical one, because if they in fact decide there's no standing, that's the only issue left to go to the Supreme Court, because it says the court has no competence to hear the case, the appellate court, because there's no plaintiff who's brought a matter that is within the court's purview under the Constitution to have an actual case or controversy. And so that would be an interesting, to say the least, outcome.
Yeah, so just to summarize 2 points there, Kevin. One is the argument that Kevin's making is on this word isolated, you can interpret it one of two ways. You can interpret it the expansive way that I tend to, which means you'd be infringing. But he's saying if you do interpret it that way, these claims are in trouble on other grounds. They're going to flunk. They're going to be declared invalid if they're ever challenged. That's basically the content of that argument. And then the upshot of the stuff about standing is we may never get any further than we've already gotten in this case on the specifics of the case. This one residual uncertainty about can you patent. It could be that we're not going to get there because it will be decided now on the basis of standing. That is, it no longer matters, and this case is not about an active controversy.
So the only point I'd make is that I agree with Kevin on the isolated DNA, but I don't see how that gets you out of the 15-mer box. I think the 15-mer box is still there.
I think it's the same answer. And Bob, actually, it's funny because we thought we were smarter than we were back in the day. And the idea was, although it should have been 16-mer, if you do just statistics, a 16-mer should occur once at random, once in DNA every 4 billion bases. And so it should be unique. The problem is that DNA is not random. It's a historical molecule. And so a lot of these sequences where you can pick out 16 mers, which encode 5 amino acids plus 1 base, they're in common. These little pieces of protein are in common in lots of proteins. So as Bob showed in his paper, if you actually look, some of these 15 mers are found all over the chromosome. And so there's no specificity anymore. And so they're invalid for that reason.
Right, but it's very difficult to do that work at the PTO level, let alone at the. I mean, it's much easier and crisper to say that gDNA is unpatentable, which is what Bryson was getting at in his opinion. So, then you get to administrative costs. What administrative costs are you going to impose upon the courts or the PTO?
You could always
So
impose
I
it on
don't
the
mean to
applicants.
interrupt this discussion.
we're having
Yes,
fun.
you do.
We're having fun.
Yeah,
But I
yeah.
do.
Yes, you do. Yeah, that's
I just wanted
perfectly
to
fine.
give
That's
to
great.
give the folks who
Sure.
are here a chance to ask questions of you all. We have a microphone that we can pass around, and I would just ask you to give your name and your affiliation.
I know, I think it's not, yeah.
I can start it off. I'm Derek Scholes at National Human Genome Research Institute. I'm curious about this disagreement or differing position between the USPTO and the Department of Justice. I guess my question is two parts. Is it we're seeing it through the eyes of gene patents, but is this something that you see in case law all the time, that the patents have been granted, but the Department of Justice ends up taking a different position? And then the second part is this a disagreement between the USPTO today and the Department of Justice today, or is it a disagreement between the USPTO 20 years ago when more of these patents were being granted, and the Department of Justice position in 2011?
So it's very rare to see these sorts of disagreements. I'm not aware of another case involving the PTO where a policy that the PTO has put forward was explicitly rejected. Not that the PTO has rulemaking authority, because it doesn't, but generally speaking, the Justice Department defends the positions taken by agencies. The curious piece in this puzzle is that in the view of the Department of Justice, the PTO isn't the only stakeholder, that all of those other agencies that I read out are also stakeholders, and equally so in the view of the Department of Justice. Whether it's a disagreement between the PTO of 20 years ago and the current Department of Justice, that may be, it is the case. Bryson makes this point, and it's, as a lawyer, that's absolutely right. The executive branch is unitary. You can't have multiple voices in the executive branch, and the Department of Justice, under congressional statute, has the sole authority to represent the executive branch in litigation. No other department does. Now, oftentimes the Department of Justice will let the PTO represent themselves before the Federal Circuit, but that's a matter of their discretion. So I think I'm a fairly starchy administrative lawyer, and I think the executive branch has to be unitary. So at this point, it has to be a disagreement with respect to the PTO of 20 years ago.
Although, Judge Moore did point out, both at oral argument and in her opinion, that she sees a clear dispute within the administration. And although I think Artie's right.
Well, Judge Bryson suggested that, but with respect to Judge Moore's opinion, I don't see how you can say that the executive branch has two heads. It doesn't. It has one head, and that's the president.
Sure.
So it would follow from that then that if Myriad filed these claims today, assuming that the USPTO and Department of Justice are aligned with each other, that they would fail, right? They would be rejected.
Well, there's a tricky
Or not.
question as to whether the PTO is going to be continuing to operate until this decision is resolved, this issue is resolved under its old policy. That
Haven't they been? I mean,
Yeah,
Gene Patton's claims come out every week. So.
right. So they are saying that they're continuing until the issue is resolved to operate under their old policy, which I take it the Justice Department has also signed off on. So it's a that's, I take it, the compromise that's been negotiated.
So Derek, your question though raises another point that we discussed on our telecom, but we haven't raised here, and I'll just raise it because your question elicits it. If this were happening now, number one, the way these claims are drafted would be very much more precise, because this was state of the art in 1994. But there's all sorts of stuff that's happened in the meantime that would change the way the claims are framed. So it would be a completely different debate now. One of the other differences would be that as of Friday, when the president signs the bill, there will be an opposition proceeding, a post-grant proceeding that isn't litigation. It's short of litigation, but it's going to involve, you can challenge a patent for a period after it's been issued. And this case is actually interesting in that respect because Europe has that authority now, and it happened in this case. So what happens when that happens, when somebody challenges it? And what happened was it was the same patents that have resulted in U.S. patents were the ones that were the starting point for the European process. And at the end of this opposition process, the European Patent Office did grant patent claims on the molecules, but they were narrowed to the mutations that had actually been fully disclosed and kind of characterized in the specification. So it narrowed the range. So the claims that we've been discussing today would not exist in their current form at all. They would have been much narrower. But there would still be some.
These particular patents can't be re-examined because
Right.
post-grant review is
I'm
only
sorry,
for a year.
yes, thank you for
Yeah,
clarifying
yeah.
that. So this is a counterfactual.
Perspective.
But this is what might happen now going forward with a similar case.
But if we've learned anything from those 20 years, we'd expect, and I think it is the case, that the grounds for opposition would be much narrower as well, because you're not going to have these same sorts of claims today.
So did we have any other, gosh, we didn't mean
We
to
were not
suppress all,
provocative.
we
Or maybe
weren't
it's
nasty to each
exhausted.
other enough up here. Yeah, Joe?
Jo Boffman from the American Society of Human Genetics. If I accept that the full genome sequence, the extracted DNA sequenced fully, and the clipped portion that has been sequenced in this patent, if in fact the full genome sequence does not infringe on this patent, could you make a comment about the process of microarray diagnosis, where in fact several or hundreds of clips of DNA are put onto a single plate, and then those clips are read? And the question that I'm really trying to get at is, if the 15 mer of importance here is on that plate, if I read that 15 mer and give it to the patient, have I infringed on the patent?
Making the array would, if, and Bob points out properly that and Arti, that the current 15 mer claims are invalid. However, if you were to have a claim that specifically recited a 15 mer that comprised a mutation that was unique, so that it wasn't all over the genome, and so if you had that, and if you did in fact make that chip, the infringer would be the person who made the chip, first of all, although you might be an infringer as well by using it. And that does raise the one thing I think that we probably all agree with, is that there is this potential for what traditionally has been called a patent thicket. that you would have this problem of if you want to have a chip with 10,000 genes on it, you have to pay 10,000 people their royalties. And that is probably the real problem at the base of this. It's a commercial problem because it's a problem with people making the chips, but obviously there's an access problem there as well, people who want to use the chips. And the only thing you can say to that is twofold. One is that for the broad claims, most of them, Myriad's claims expire in about 3 years. And the claims to most human DNA will expire in about 2020 because most of them were filed in about 2000. So that part won't be the problem. It still may be a problem for the specific chip, and all I can say is that there have been plenty of other instances and plenty of other areas where you've had this problem, and it's been resolved in the industry because people want to make money. And so if you have this block, eventually you'll have some, whether it was in the computer industry, in the telecom industry, they have standard setting. In music, they have ASCAP and BMI to do it. There are ways around this that we should encourage, that should be facilitated, but that's probably the place where there really is this issue of could the patents get in the way of the development of the technology? And the answer is yeah, maybe. Maybe.
And I think that's what Judge Bryson is concerned about, quite frankly. You know, the example of telecom and software is not necessarily an inspiring one.
Well, but it's worked. It may not be perfect, but it's worked.
I mean, it depends on
My
your
cell
perspective.
phone works
Well,
most of the time.
So I know
Some
for
places.
a fact that people are quite concerned, including cell phone manufacturers, about what's happening with smartphone patent litigation at the International Trade Commission, where you can get exclusion orders for infringing a patent, not simply damages, but exclusion orders.
So there's a layer of where your questions take us that is also very imponderable because we don't quite know what the business models are going to be and stuff like that. Because a lot of how this stuff plays out in the real world really depends on not only that people want to make money, but how they're going to do that in detail. So there are companies across the board who are really interested in next generation diagnostics, full genome sequence, all these microarray technologies. That it could well be that two things would be true at the same time. Is number one, they would be infringing in some way. And one thing that you can do, like going all the way back to sewing machines, right? Mm-hmm. You do have these situations where there's a logjam, where nobody can go forward unless you aggregate all this stuff, and you get this problem of a thicket. And you can solve that problem. Sometimes it takes a really long time to do that. Aircraft manufacturing, broadcast radio, sewing machines, we've got instances. And sometimes a really messy, nasty process that takes about a decade to sort out. And nobody wants that to happen, but that is one way to solve the problem. The other way, and this is one reason I think this case was brought, is one way is just get rid of the thicket. Just cut right through it. It's the Gordian knot, and just cut it. So that's the policy debate that I think is going on, and you see people breaking into factions on which of those policy choices is the right way to go. And that's part of what's at stake here.
Of course, the thicket has consequences. Cutting the thicket has consequences.
Yeah, it does.
And that's where I really do think if that's the way people do it, then all of this stuff is going to go into the mystery chip that you can see what's on it, but you don't know what's important because I've encrypted it in a way that I know what's on it that's important, but every chip is different, and every position, and my computer can figure it out, but you'll never figure it out because you can encrypt these things in a way that you'd have to look at a billion chips to figure it out. And so that way would mean that your chip, it wouldn't take a decade, it would be perpetual. Somebody else would have to figure out exactly what you did, and there'd be an incentive for you not to disclose it. So I think that's really probably the wrong way, and anytime you have these sort of Gordian solutions, there are both pros and
there are
cons.
secondary
So-
consequences. And
So what's happening with software and telecom, with which I'm also familiar, is there's a lot of so-called independent invention going on, and the argument is, well, at least for those situations where we have thickets, but the person who is claiming to be the infringer, who is claimed as the infringer, independently invented, we should have an independent invention defense. That's another way of addressing these issues that Congress has thought of, but has thus far, at least in most circumstances, not endorsed.
Although the prior user rights gives a certain amount of
Prior
that.
user rights gives a little bit of that, but in limited context
Right,
as well.
but keep in
Yeah.
mind, the reason that we don't do it is because we want to encourage disclosure. And if you invent and keep it secret, and then you can profit from it, and the rest of us don't. And that's perpetual as well. So the reason that there's been a resistance to that is to encourage people, if they want protected rights, that they have to disclose, because then those rights expire.
So Kevin gave one rationale for why you might think that patents would be a good thing in this area, and that's because the alternative is trade secrecy. The other reason that is offered as a reason for thinking that patents might be valuable is that the direction that diagnostics is going is it's getting pretty complicated and expensive, and it requires induced investment by the private sector. So what you get if you can somehow protect something through patents is you can induce private R&D that might not otherwise be available. That is, you induce that investment in developing the technology post-discovery and turning it into a product or service that's available on the market that may not happen without the patent incentive. So that's one of the things that's at stake. The counter-argument to that, of course, is, well, diagnostics, there are lots of other pathways to getting those, and they don't involve private investment. But that's a secondary consequence of using the Gordian knot solution, which is you are, to the extent that patents are socially valuable, you lose that additional incentive for private R&D.
It's actually funny, Bob, because we talk about the tragedy of the anti-commons because it's a clever phrase, but where it comes from is the tragedy of the commons. That been in England, if nobody had responsibility for the common land, nobody maintained it. And I would argue that you could develop that in diagnostics. If you basically said that you couldn't protect them, then who would have the incentive to do all of the genetic counseling work and all of the payer work and all those things that need to be done to move a discovery into actually a useful diagnostic? Even though everybody could do it, there would be less incentive for each individual provider to do it, because if they got a payer to do their test, that would be for everybody else who's offering the test. So you wouldn't have that incentive as well.
And to throw yet another spanner into the mix, there's the question of whether the FDA is going to regulate all this. And FDA regulation creates a need for patents on the one hand, because you need more investment in order to need to invest more money in order to get through FDA regulation. On the other hand, the fact of FDA regulation also creates barriers to entry by competitors. So it's a double-edged sword with respect to all of these issues.
Well, and it also opens the prospect of creating a zone of exemption from infringement liability, because there's a built-in in the
Well,
Boehner
there's-
amendment.
yeah,
So
there's
it
the
makes it
Boehner
really, really
amendment. Yeah, it
complicated
makes
if
it
FDA gets into the
extremely
game.
complicated.
So look, all these domains of law, antitrust law, patent law, and regulatory law, all interact in really complicated ways in this domain. We don't need to take you into the inside baseball.
Yeah, I was actually a little surprised that the FDA wasn't listed as one of the agencies
Yeah,
that
it
had
was
an
interesting.
interest in this case. Maybe that was the among others.
Yeah. So are there other questions for us? Yes, there are a couple in the back
Good.
there.
What about the issue of that patents basically create a monopoly on a diagnostic in this case, so that the women are basically they have no one to turn to for secondary assessments, that sort of thing.
Well, yeah, I think Myriad would have been very smart if they would have licensed somebody else that, with the caveat that you got a second opinion only if you came in with the Myriad report. So they had gotten their, that it was really a second opinion, you weren't trying to circumvent. I think that there is a study that is, we talked about Debbie Wasserman Schultz's amendment that was pulled. Actually, I don't know the inside baseball part, but I do know that the ACLU and many of their friends wrote a scathing letter to Congress that says, do not pass this amendment, and that was at least part of the motivation why it was pulled from the patent reform bill. But there's a study that's going to be done to look at that question of whether there should be some second opinion sort of a provision. And it seems to me that if you're a patentee, there's a principle of patent exhaustion. If somebody uses your service and you've gotten whatever the royalty is for that, then a second opinion isn't really that much of a harm. The argument, of course, is twofold. One is that they don't do it correctly, and so raises this problem of there's been a mistake. And secondly, that they use it to circumvent the patent holder in the first instance. But absent that, I think there's a place for it. At least we'll find out if there's a place for it when the study's done.
So apparently, one of a little bit of the backstory was, at least on the floor of the House, there was a discussion about the need for empirical evidence on this question, and that's, I think, why the requirement for a study was put in. Unfortunately, the PTA only has 9 months, which is not a whole heck of a lot of
So
time.
unless the evidence already exists, it's going to be hard
Yeah.
to find. And I think if there were empirical evidence, we would know about it by now. So I think the USPTO is in kind of an awkward position trying to respond on this time frame on this question, because I think it's going to be hard to find that evidence. But that is the issue. It is the verification testing. It's a real issue when you've got a monopoly, right? It does put the patent law in a privileged position in determining the medical practices. Yeah.
So what kind of evidence would you be looking for?
You'd need to look for real cases where it's come up. Two of the plaintiffs in this case, that was an issue. And I'm sure that those cases will be examined in greater detail in the wake of the fact that Section 27 is part of the law. So it has to be done. So thank you for that question.
I would mention, while the mic's being passed, just till it gets to you, that remember, part of this is a U.S. healthcare system problem. It's an allocation of resources problem. It's an insurance problem, because I don't care whether it's patented or not. If you get something that insurance doesn't want to pay for, they're not going to pay for it. So we can't try to make a patent solution to an insurance problem.
Although one of the reasons that so many different branches of government are interested is because the U.S. pays for a lot of that insurance.
Yeah, the other agency that really has a huge stake here that isn't on that list is the Center for Medicaid and Medicare Services.
CMS, yeah.
So the U.S. pays for about $887 billion of health services. It's the single largest payer of health services in the world by far.
Right.
And so we've got a direct stake in that. Yes. Your question.
Sure. My name is Alice Popejoy. I'm with the Association for Women in Science, but here out of personal interest, not affiliation. So I have two. Well, first of all, I was wondering if you could sort of address the isolation as changing the molecule. How is that justified? That if you just isolate it, that's in effect changing it? And then is there an argument to be made that you can't prove that those 15 mers are not found in nature? Like, if we don't fully understand the mechanism for how the whole genome works together, which as I understand it, we don't, could you make the argument that it's possible that even if you change successfully a sequence of DNA, that in fact it could exist in nature?
Well, it's hard to prove a negative, right? But I think that we know where we get genes from. We get them from chromosomes. And one of the arguments that people make is that you see a change when you actually make something useful. And the gene is useful for making a protein in your cell, but it's not useful for making a therapeutic, for example. So the traditional argument has been that isolating the DNA gives it a new utility. First of all, it's very back, and remember, a lot of this stems from the technology when we didn't have the Human Genome Project's results, and where the technology of isolating the genes was rather difficult. The idea of having this gene in your body that you knew worked because it made interferon, erythropoietin, TPA, what have you, was fine, but it didn't do very much good. In fact, there were patents on people who extracted EPO from urine because it was the only way you could get it, as opposed to actually cloning the gene and having a cell make it. You've now changed the utility by isolating it, and that was enough to make it, I think the theory was that was enough to make it patent eligible. In terms of the 15 mers, that was one of the arguments that was made by the ACLU in its petition for rehearing, that during the process of DNA synthesis you make Okazaki fragments, which are small pieces of DNA. But I would argue that, again, very similarly, they're not isolated. They're in the cell. They're not useful except to the cell. And so I think that there still is a basis for claiming them, except if they're not novel. And that's where the random 15-mer isn't so random, and that causes patentability problems on that side of it. So,
And just an embellishment on that, the fact is, in my reading, those claims would be infringed, but they would be invalid. So I don't know where it would go. And what is at stake? So we've had 4 judges that have addressed the first part of your question. What's this isolation stuff really mean? And you have, there's a lot of disagreement still. It's very clear that we don't quite know what it means. And it would be really nice, actually, to have some precedent set in case law so that we begin. We've got much more now than we had in May of 2009. It's actually one of the social utilities of litigation, is you narrow zones of uncertainty. And I think a lot of that has already been happened, but the place where it still exists is we don't know what that word means. And you have 4 judges at the federal level who have spent a lot of prose trying to figure out what it means, and they've used 4 completely different lines of argument. There's not all 4, Moore, Bryson, Laurie, and Sweet have all come at that question, and they've used different trains of argument to reach their respective conclusions. So not only the conclusions differ, but the justification for those conclusions differ, and that means that we don't quite know how to answer your question, which means the question was great, but we don't have an answer.
Let me just add one caveat to that, though. There's a axiom in patent law: the patentee can be his or her own lexicographer. And
So do that again.
Yeah, because I'm sure they really want to hear it.
it's such a sexy axiom. But the point is, it's applicable in this case because Myriad did discuss what it meant by isolated, and I think this is where, I hate to disagree with my colleague Bob, but I think
She
Kevin,
really hates it, you can tell.
I think this is where Kevin is right. They're pretty clear in what they mean by isolated, and that's kind of pretty much at least gene level.
And it's why everybody really there is a one of the things that the Federal Circuit has set out. How do you construe claims? How do you figure out what they mean? And the absolute rock bottom first principle is, what does the claim say as it's understood by people who do this? And I would argue that if you look at how, at least in patent law, which may be different from science, but in patent law, isolated means, you have the piece of DNA that encodes the gene with maybe a little bit at the edges because of this infringement issue, but you don't have the whole chromosome. So it would be inconsistent with all of that way it's used by everybody else to say, no, it can be read broadly enough if you look at it correctly to encompass, say, whole chromosomal DNA. So. Let me actually add one thing to what Bob said very briefly. There's a fifth opinion about this that has nothing to do with gene patents, but it comes from the Chief Judge of the Federal Circuit.
Oh, yeah.
There's a case called Claussen that just came down, and it was about a way of determining the best way of immunizing a patient to minimize the occurrence or the severity of immune-related side effects, disease. And that it has nothing to do strictly with this case. But as well as the majority of the opinion and dissent, sort of very rarely, I think, the Chief Judge took the time to file a concurring opinion, which he called additional views. And in those views, his point of view is that 101, the section of the statute on patent eligibility, is the wrong test to be using. He calls it a coarse filter. And it maybe relates to this Gordian solution, that it's too broad, and making these distinctions have ramifications that go way beyond the case that's in front of the court. And I think I've alluded to it, that the rationale for saying that something isn't patent eligible for genes is going to spill out to antibodies and proteins and a lot of other naturally occurring products, and may give us consequences that we don't want. But I recommend, if you have nothing better to do before you go to bed tonight, read Judge Rader's opinion, because it's short, but it does give a policy view, and he uses biotechnology as an example of an industry that would have been hurt had we had this discussion 20 years ago.
So, and just to double click on that, Judge Rader's, just truth be told, in full disclosure, this is actually a point that probably all three of us agree on. So what's going on in this case is if you're going to get a patent, you have to cross the threshold to say this is the sort of thing that can be patented at all. And that's one of the things that Section 101 of the patent statute does. It says there are these four kinds of things you can patent: process, composition of matter, the two that are relevant here. So the DNA molecule is a composition of matter, and that's what's at stake here. And that's basically the threshold that says, yes, you can look at the other stuff. Have you met the criteria for, was it obvious, is it new, is it fully disclosed, is it described adequately? All these other criteria about whether you should get a patent or not, you don't even get to those until you've crossed the threshold to say this is the sort of thing that can be patented at all. That's what this case is argued on so far. It could have been argued on these other grounds, in which case you might get the same outcome, but on very different reasons. And so I think probably, I don't know, Arti, for you for sure, but I know Kevin and I are on the same boat on this, but please understand, not everybody on the planet is. There are many people who are not in this room who would say, no, 101 is the right way. DNA is the sort of thing that should not be patented at all. So that's at stake, and the three of us kind of agree that maybe it'd be better if we allow it in the door, but then we use the other doctrines of patent law to winnow the forest so that we don't get as thick of a thicket as we otherwise would. But that's kind of where we are. So just understand, we're kind of in unison on that particular point, but there's a whole wide world out there that doesn't agree with us.
So I agree, Bob, that 101 is a very coarse filter, and in general, the patent law types don't like 101 because they see it as confusing and metaphysical and all the rest of it. And I think that's all, I agree with all of that. The only argument I would make, which I don't necessarily believe, but just to put it out there, is that some people think it's administratively easier to use
Yeah,
than
it cleans
the other.
things. It's a
Yeah.
filter,
Yeah.
right? So you
Yeah.
have filters when you're trying
Right,
to keep
and
stuff
so-
from getting into your system.
Yeah, and that's, I think, for those who have legitimate arguments with respect to 101, that would be the best one.
Yeah. So have we worn you out? Are there any further questions? Yeah.
Thank you. I'm Tom DeLinge. I'm with BIO, the Biotechnology Industry Organization. My question goes back to the whole genome sequencing issue that we talked about a bit. Our association represents companies that do therapeutics, diagnostics, whole genome sequencing, or developing those kinds of tests. They patent DNA molecules, patent all sorts of things. None of our members, even the ones that are doing whole genome testing, have argued against the patent eligibility of DNA molecules to us. They've actually said that they support the continued patent eligibility of them. They are concerned about this kind of potential thicket, if you will, of having to figure out all of the patents that are out there that might infringe or might not infringe. And they are trying to develop their own kind of commercial practices of dealing with that. But we see a lot of investment going on in that area, despite the fact that there is this potential uncertainty. And the companies that are doing this work, that are bio members, tell us they're doing it, and they're raising lots and lots of money, and they're actually patenting their inventions, and they're proceeding. So I'm just wondering, is there any kind of evidence that this potential thicket out there is actually impeding the development of these types of whole genome tests? And if not, how do you kind of get that evidence? Because before you untie the Gordian knot with this very coarse filter, it would seem you'd want to make sure that there's a real problem that needs to be addressed.
Yeah, so that's, that's this isn't the $64,000 question. This is like the $64 million question, right? And you're absolutely right, Tom, that there's a lot of commercial investment activity going on. Your question really gets at how much would be going on, but for. I have to say that the investors that are in this space are fully aware of this risk, because we talk to them and they are conscious of it. So whatever whatever you say about markets, they account for risk and stuff like that. So they know that this is factored in. Would there be more investment if there weren't this shadow of uncertainty? I don't think it's a very big uncertainty, frankly, because there are all these ways of solving this problem. You could have an ASCAP model. A lot of if it is worth fighting over, the claims, we will begin to get case law because people will sue each other. And then we will have case law and we'll know how to interpret them and we'll know what the rules are. So there are ways to solve the problem. Some of them are really messy and expensive and slow, which is not what any of us would like. But there are solutions. And so right now, I don't know how many companies are worried enough that they aren't doing it. I've talked to some people that claim that they would have but aren't. But I've also talked to a lot of people who are aware of the risks and are plowing ahead, and I don't know how to quantitate that. And I frankly don't think probably anybody can. So the other answer to that, and I'll here turn to you guys because you actually deal with the real world and many other kinds of places where this has come up, is this is not unprecedented in other areas. You do have thickets, as we've alluded to, and it does happen. Predicting the risk in advance, I think, is really, really hard because you're trying to predict the risk of the companies that are making the investments, and you're trying to predict the risk coming from a very fluid area of jurisprudence and patent trolls and multiple licensing agreements. And how are you going to predict who's going to sue whom in advance?
So the thing that would worry me, and this is really based more upon software and telecom than upon anything that's happening in genetics, is that the big players, I think, will plow ahead because, look, I mean, if you're Google, you can plow ahead and then pay for $4.5 billion, I think, for Motorola to get their defensive patent portfolio. You can do that, that's just chump change. What worries me, and this there's some evidence of this in the software and telecom space, is that there is evidence, Ian Coburn has done some good work on this, that small firms, there are barriers to entry in certain areas of software as a consequence of perceived patent thickets. And this is not just interview work. This is based upon kind of econometric data controlling for lots of different variables. Why do certain firms not enter into certain software spaces?
There actually may be a very simple scientific answer that your question actually may gave me a thought of. You know, what if it's the case that your oligos that identify specific mutations can be 15 bases long? And what if the case is that none of them are novel? Because at that level, they're just not. Okay, then they're not, then they don't infringe. And I know Arti says for the PTO it would be hard, but trust me, it wouldn't be hard for a company to figure that
Yeah,
out. That's
yeah.
easy. And
Yeah.
so if you look at these mutations and look at the disclosure from the patent specification, which you can freely use to do this, and you're not infringing, and if you look at that and identify your 15-mer and find by interrogating the Human Genome Database that it occurs 300,000 times, or whatever Bob's number was, then you can use that pretty much with impunity. Doesn't mean you're not going to be sued, but means you have a summary judgment motion that says, you know, Judge, it's undisputed that this is present 300,000 times in the human genome and was put on the Human Genome Database in 1998. Done, okay? So that may be, the length may actually be the key to being able to make these sorts of chips that wouldn't be infringing. And we'll see how that plays out in terms of how you then protect it, but that may be the answer.
Yeah.
So again, great questions, and thank you all. Thank you all for coming. I don't want to suppress any questions, but we've done this twice, and I don't want to keep doing it. So thank you all for coming.
Thank you.
Thanks.
Thank you. You got- Thank you, Arti.
Thank
Very
you.
nice.
This is
Wonderful.
very
Excellent.
interesting.
Bob, excellent. Very nice.
So,
Very nice.
I mean, I do think the big players will have.